Thank you so much for the introduction, and thank you to the LSI team for organizing this conference. I had a great time here, despite the rain. I'll tell a little bit about the story at Invenio Imaging. The NEO laser imaging system uses laser spectroscopy and artificial intelligence to analyze fresh tissue specimen directly in the operating room. Such real-time tissue analysis is a big unmet need that impacts over 13 million procedures every year. The NEO is already FDA registered and CE certified. We've placed about 12 systems in the field so far and completed over 3,000 cases successfully. The primary focus of Invenio for the next couple of years is a study that we're doing jointly together with Johnson & Johnson to develop the first clinically approved artificial intelligence to detect lung cancer in the operating room. So there are three major trends in lung cancer diagnosis. First, the screening with low-dose CT allows to identify early-stage lesions in high-risk patients. Then robotic bronchoscopy allows to biopsy those specimens very non-invasively. And last, the next-generation sequencing is then applied to the sample to identify the key driver mutations that then allows to triage patients to targeted therapies. However, there's also a sad fact that only 35% of patients that would be eligible for such a targeted therapy, in fact, obtain such ideal treatments. The main reason is that a lot of these biopsy procedures are non-adequate for identifying or obtaining tissue that then can be used successfully for sequencing. The primary reason for that is that existing methods for rapid on-site evaluation, or ROSE, are underutilized because they are inconsistent and slow. Another reason is that very often those techniques cannot identify whether a specimen is indeed a good candidate for next-generation sequencing. At Invenio, we developed the NEO laser imaging system that uses laser spectroscopy to image a tissue biopsy. We use the spectroscopic information to then apply a digital stain to the image that is chosen to mimic traditional H&E staining. One of the major advantages of this approach is that it's non-destructive to the specimen, such that it can then be used to be analyzed by histopathology using traditional methods and next-generation sequencing. Another advantage is that our sample preparation is trivial. We have a proprietary consumable that we call the NEO slide that can be used by the nursing staff that's already in the operating room to make the sample preparation and then image it directly in the operating room. So this avoids having to send the tissue specimen into a dedicated frozen section laboratory where it's then stained, sectioned, and then ultimately interpreted by the pathologist. We are combining this technology with artificial intelligence, where we're training algorithms to distinguish samples that are suspicious for cancer versus samples that are not suspicious for cancer. We have demonstrated in a large study that we can do that very successfully, non-inferior to pathologists using traditional methods. But we can take it a step further. We can go beyond saying there's cancer present in the specimen or not. We can actually identify how much cancer there is, and that is one of the key indicators for next-generation sequencing. We have some exciting work going on in the lab that's not clinical at this point, but we are showing that we can go beyond detection of cancer to actually identifying what the key mutations are in the specimen. This uses purely morphologic features instead of sequencing. It's therefore rapid and for the first time available at the time of surgery. This becomes very exciting as we think about the future of bronchoscopy, where the pulmonologists are trying to combine diagnosis and treatment in a single procedure. There's technologies in ablation, there's like local delivery of chemotherapies. Having accurate diagnosis in the OR will be key enabling for these applications. So looking at the market in total, there's about 13 million procedures that are impacted by intraoperative tissue analysis. Lung cancer by far is the largest opportunity for us. This is because there's 1.5 million primary nodules identified each year in the US alone. So at a modest revenue, this provides a $1 billion opportunity in the United States alone. The imaging system has been commercially available for 2 years. We have sold about 12 systems, 3 of which in the European Union. 3 of our existing sites have gone to purchase second systems where they want to use them in different applications. We have many happy customers that have been working with us in various applications. Altogether, we've completed over 3,000 cases successfully. We launched the technology actually in brain cancer, but then quickly expanded with the existing customers into all these various applications. What we have found is that our imaging technology delivers image quality that's superior to any of the other competing technologies. This is particularly important for applications where cytologists use this tissue level or subcellular information in order to make a diagnosis. We believe that's not just a nice to have, we think that it's image quality that ultimately drives the accuracy for diagnosis in the operating room. We have a large patent portfolio and a growing proprietary data set that we use for our machine learning. As we commercialize this technology, we apply a hybrid pricing model that consists of the capital sale, a manual service and maintenance plan, as well as our proprietary consumables. We are achieving these prices in the US with our customers today, and we believe that as we move into the lung cancer space, where procedure volumes are high and ORs are booked 3 or 4 times a day, and multiple specimens are taken per procedure, it will be largely the consumable revenue that drives our revenue. As we try to sell this, we always start with the surgical champion, but then use an incremental procedures model to convince the hospital administrator. This is justified because hospitals really benefit from having downstream procedures like thoracic surgeries or SBRT that can only be prescribed or prevented if a definitive diagnosis has been obtained. So non-diagnostic biopsy are a real issue for hospitals. The neuroimaging system is already FDA approved. It's CE certified since 2021. We also have a CE certified AI algorithm in Europe for brain tumors. But the primary focus of the company right now is to get the first FDA clearance for lung cancer. For that, we've been planning on a study that we call the on-site study that will enroll 900 patients prospectively. That's possible because this is like such high volume procedures. It's jointly with Johnson & Johnson and contains about 3 study arms for the 3 different biopsy instruments that are used in those procedures. The primary endpoint will be to obtain a non-inferiority diagnosis compared to the standard of care, that's ROSE. We have a great team. My co-founder, Jay Troutman, he's the CEO, and he has had multiple successful exits in recent years in the biotech industry. We also have great support from clinical, regulatory affairs, R&D, as well as manufacturing. So as we look at our commercial timeline, we've met with the FDA and they've provided a lot of feedback to our proposed clinical study. We are going through those changes right now and expect to enroll the first patients in July this year. As I mentioned, there's like 3 different arms to the study where we expect to hit the first endpoint for forceps biopsy in the middle of 2024 that we then use and to obtain like de novo clearance from the FDA. We have a ongoing sales, so we anticipate that we have about $1.4 million in sales from the imaging system this year as well as next year. Our sales force is small, but we plan to really launch in the lung space as soon as we have the clearance. With that, thank you. I'll be outside if you have any more questions and look forward to talking with you.